Okay, so now we're going to look at this single gene genetics disorder. So we are going to look at 4 important diseases plus 2. The 2 diseases actually are not in this syllabus, but in the next 2 topics. Okay, so a single gene disorder is a result of single mutated gene. Because of one gene that mutated, we look at that particular what call it, the particular points where mutation actually take place. We have about 4,000 human diseases are caused by single gene defects. So, but what we're going to look at here is we're going to look at the sickle cell anemia. This one's quite we have learned before because of this HBS. Okay, so this one's we always have gone through. We have gone through quite many times about this sickle cell anemia. Is because of the base substitution. Okay, so we're going to revise it again. So we do have the cystic fibrosis. Okay, when we talk about cystic fibrosis is because of the base deletions. Okay, because of base deletions. And then we do have the phenylketonuria. Okay, oculocutaneous albinism, hemophilia, and also the Huntington disease. So hemophilia, oculocutaneous and phenylketonuria, these 2 actually is caused. Okay, these 2 they are caused by these 2 are caused by this what we call the mutation result in the recessive, because of homozygous recessive. Hemophilia because of the sex link. We're going to link the sex link, or we call X link. And then Huntington is a bit special here because the Huntington's is caused by the dominant allele. So Huntington disease actually is very scary in this case, because why? We just need one copy, okay, of the allele to get the disease. And the scarier part here is because the disease is a late onset disease. Okay, what's mean late onset? Means that by the time the person actually know that he or she got the Huntington disease, he or she may already in the reproductive age. So you pass down, so you pass on the allele to the next generations already. Okay. So we start the first one. Okay, we start the first one with sickle cell anemia. So because of the base substitutions, this one you have to know. This one you have to know, okay, because question always asks, okay, what are the main cause of this sickle cell anemia? Because of base substitutions, okay, where they're going to have it, where have it at the beta change, okay, because of beta polypeptide. If you look at the original one, we have the first position, second position, the third, number 4, number 5, at number 6 position is the glutamic acid. So glutamic acid is a hydrophilic R group. Okay, glutamic acid has this hydrophilic R group. Okay, but in some people, the base sequence CTC is replaced by CAC. So single base substitution. Can you see that? Adenine replace the thymine. So therefore, at the 6th position here, no longer glutamic acid, but now change to valine, which is a non-polar. Are you clear? Okay, so non-polar amino acid, or the R group. So therefore, because non-polar, it reduces the solubility of the beta globins or beta polypeptides. So that's why precipitations take place and make the cell become the sickle shape. Are you clear? Okay, so you can compare. Now, guys, when you talk about wild type, always remember, in bio, wild type basically means the normal. Okay, mutant basically means that mutated. Say the mutant cells, sickle cells, beta globin. So you can see that original CTC now become CAC. So instead of glutamic acid, now we get the valine. So what happened here is under low partial pressure oxygen, the hydrophobic side change of the valines, hydrophobic R group or the valine, at the 6 positions cause the hemoglobin molecule to aggregate to form a fibrous precipitate. and resulting in the sicklings of the red blood cells. And repeated episode of sickling damage the cell membrane, decrease the cell's elasticity. So therefore, the cell fail to return to normal shape when the normal oxygen tension is restored. So therefore, these sickle cells were going to clog at the vessel there, a small vessel or small capillary. So it will cause the vessel occlusions of ischemia. Vessel occlusion means the block. Ischemia means the lack of oxygen. Okay, so you can see that what actually happened here. Can you see that? Okay, the original ones, they have the elasticity, so they can squeeze through the small capillary. But when it comes to sickle, because of the precipitate, can you see that they form fibrous precipitate? So because of this fibrous precipitate, now they clog, can you see that, block at the blood vessel or this small blood capillary. And what happens here? The side or the cells, they cannot receive sufficient oxygen. Can you see that the blood flow won't be normal anymore? So, but at this moment, still not anemic yet. It's not anemic yet. Why? Because as long as we do have supply enough blood or supply enough oxygen, then it should be okay. But the actual anemia is caused by the hemolysis, because the what we call this the life, okay, I mean the durations, okay, the life, okay, of the red blood cells normally is about 120 days. Okay, it can functions, but for the sickle cells, because elasticity problems, the membranes lose the elasticity, hemolysis take place, it may last about 20 days. So means that the bone marrow attempt to compensate by produce more red blood cell, but it does not match with the rate of destruction. So means that the cell lies much more faster than we build or to make a new cells. Okay, so this is the first one, sickle cell anemia. So caused by what? So summarize here the summary: base substitution. Because of base substitutions, then it happened at where? It happened at the gene that code for the beta chain. Okay, so at the 6 positions, glutamic acid now exchanged by the valine from the hydrophilic R group becomes non-polar R groups, therefore reduce the solubility of the hemoglobins. So what will happen if we reduce solubility? When the oxygen partial pressure is low, then it forms this aggregate, means join together. Right? Instead of molecular base, they become joined together, it form the fibrous precipitate, cause the sicklings of the red blood cell. So the repeated episode of sickling damage the cell membrane, decrease the cell elasticity. So it means that the cell fail to return to the normal shape even though under normal oxygen tensions. Okay, so these rigid blood cells may block at the narrow capillary, leading to the vessel occlusion, mean the blockage. Ischemia means lack of O2. Okay, but if the red blood cells still enough, then it's not called, it won't cause anemics. But anemics because of the hemolysis, because the sickle cell only lasts for, can only survive for 20 days. Okay. Next one, we're going to look at this cystic fibrosis. So cystic fibrosis actually, okay, is because of this autosomal recessive manner. Okay, the disease, autosomal recessive. So what's mean autosomal recessive? So it means that 2 copies of recessive allele. Only the person will get the disease. Okay. Can I see that? Okay, so what actually happened here is we do have a special channel protein called cystic fibrosis transmembrane conductance regulator, or CFTR protein. So what is the function of CFTR? Let us look at this. Okay, now at your trachea, the cell of the trachea. So this is a lumen. It can be bronchus, it can be bronchus. So this is a lumen. What will happen here is we do have the goblet cells produce the mucus. Okay, so this is mucus. But the problem here is the mucus is thick. It's too thick. So it means that the beating actions of the cilia may not be able to remove this thick mucus. And then this mucus is going to trap the pathogen. Can I see that? Bacterial. And bacterial, because mucus won't kill. Mucus cannot kill, cannot kill pathogen or bacteria. So therefore, bacterial or pathogen accumulate. And they multiply. So then increase the risk of infection. So in order to remove this mucus, we do have in our cells, we do have these special channel proteins called CFTR. Cystic fibrosis, okay, CFTR, transmembrane conductance, regulatory conductance protein, CFTR. Okay, so what happened here is CFTR allows the chloride ion, okay, chloride ion to diffuse from the cells into the mucus layer. So when the chloride ions, okay, move into the mucus, what will happen here is going to reduce the water potential. So osmosis takes place. Then we can reduce the thickness of the mucus. Can I see that? So water molecule follow. Okay, so ecoporine. So water molecule follow. So making the mucus not so thick. But the problem is if the mutation takes place at the CFTR gene, if mutation takes place, so what happened here is no more CFTR can be available for the chloride ion to pass through, and then therefore the mucus remain very, very thick. Okay. So CFTR gene, you look at the CFTR genes, it's located at the chromosome number 7. So for information, it's about 1,480 amino acids long. So it acts as the ion channel, allow the transportation of chloride ions out of the cells. So therefore reduce the water potential outside the cells, meaning the mucus. So water molecule move out of the cell by osmosis, reduce the thickness of the mucus. This is the normal function of CFTR. Okay. So if you look at this, okay, with the CFTR, chloride ions enter into the mucus, reduce the thickness of the mucus because of the osmosis. But if the mutant CFTR channel, mutant basically means that they don't allow, for example, okay, so if they didn't allow, so mucus remain thick, what will happen here if the mucus remain thick? They trap the bacteria continue to multiply, the cilia cannot remove the mucus, therefore it increase the risk of the infections. Okay, so if you look at this type of mutations, what kind of mutation actually take place? They are found that more than 1,500 type of mutation. So it's a multiple allele case. Are you clear? But the most common one is the deletions of 3 nucleotides. Can I say a deletion of 3 nucleotides that result in the loss of 1 amino acid, phenylalines, at the position 508 on a protein. So what you need to know here, at least deletions of the 3 nucleotides. So because of these deletions of 3 nucleotides here, the remaining ones still remain the same. So therefore, there is no frame shift. Okay, it's just missing 1 amino acid, no frame shift. Okay, so this is what we call cystic fibrosis. We're going to continue the cystic fibrosis in the next 2 chapters, where we're going to look at the gene therapy, how the gene therapy may help. Okay, the gene therapy may help to treat the cystic fibrosis, and we're going to look at it how success the gene therapy have been helped to reduce the symptom of the CFTR. Okay. So the next one is the phenylketonuria. Again, this one at this moment, okay, you look at past year, a lot of questions have been answered about this phenylketonuria, but in new syllabus have been removed. So this one for your informations only. Okay. So phenylketonuria again is the autosomal recessive due to the mutation onto this PAH gene. This PAH stands for phenylalanine hydroxylase. Okay, so because of the mutation, it results in the low level of the enzyme. So it means the enzyme is very, very low. It may not be able to cut, okay, to carry out the hydrolysis process. So phenylalanine hydroxylase is responsible in the catalysis of phenylalanine to thyroxine. So in the process here, If you look at this, we have the phenylalanines. We want to convert phenylalanines into the thyroxine. Thyroxines can be converted in the pathway to form melanin. Melanin is the pigment. What will happen here is to hydrolyze the phenylalanines to form thyroxine to convert, you need the enzyme PAH. We need the enzyme called PAH. So what will happen here is phenylalanines, okay, if there is a mutation because of homozygous recessive, so means that the big A, so you will have the enzyme. So big A, small a, heterozygous, no problem, we still have the enzyme, PAH enzyme. But if a small a, homozygous recessive, no enzyme. So when PAH enzyme is not available, phenylalanines accumulate. Are you clear? Thyroxine concentrations low, therefore melanin productions will be low. Understand the pathway here, the phenylalanines. So phenylalanines increase, thyroxine decrease, melanin decrease. So what will be the consequence here? So basically phenylalanine hydroxylase or PAH responsible in the catalysis of the phenylalanine to thyroxine, then can be converted to melanin, which is the brown pigment. Okay, so but the children with PKU tend to have the lighter skin, okay, compared to the unaffected family member, and because of lighter skin, they are prone to the skin disorder, for example, the eczema. Okay, because of this pigmentation, not or insufficient pigmentations. Okay, so they look at it, it's something similar to albinism, means that it's quite wide, fair skin, okay, a very light skin, okay, compared to the normal person. So most common one, again, is a single base substitution. Okay, other PH mutations delete small number of DNA nucleotides from the genes, but the most common one is a single base substitution. But we do know that it's more than 500 mutations that have been identified. Okay, but the most common one, single base substitution. But the thing here is phenylalanines accumulation is not a good thing because our brain nerve cells sensitive to the build up of the phenylalanines. So when the phenylalanines can build up to the harmful level in the body, it can cause the intellectual disability. So it means that disorientation, because the nerve cell, they are sensitive to phenylalanine level, they may be damaged. So it will cause excessive amounts, can cause the brain damage. So it can have the delayed development, it can have behavior problem, and may have a psychiatric disorder. They are very, very common in the PKU patients. Because of what? Because of the phenylalanines level build up. Okay. So untreated individual may have the musty or the mouse light order because of the phenyl group of the phenylalanines. So how to treat? So we can't treat, we only can control the diet because phenylalanines usually appear as the artificial sweetener, right? Phenylalanines, artificial sweetener. So it means that the patients with the phenylalanines, they cannot take this artificial sweetener in a high concentration. So it means that they must adhere to a very, very special diet, particularly low in the phenylalanines, so that the brain development will be optimal. Because why? In the body, they take longer period of time to remove the phenylalanine. So if they keep on eating the foods in high in phenylalanines, what will happen will be, you can see that their mental developments or the intellectual developments will be slower because the brain cells won't be able to survive due to the sensitivity of the brain cells on this because of these phenylalanines, okay, build up. We come to the next disease, oculocutaneous albinism. This is the new topics that add in the syllabus, okay, the new topics. Again, because of autosomal recessive. Okay, again, because of the pigmentation. So if you look at the PKU and the albinism, they share the same pathway. If you look at this, can you see that? Before this pathway, we can see that. Okay, we have these phenylalanines, which involve PAH. The same pathway, but different enzyme is affected in this case. The enzyme affected here is the thyroxinase. Sorry. The thyroxinase, can you see that? So thyroxinase is the one that converts the DOPA into the dopaquinones. And dopaquinones will be converted to the melanin. So if the enzyme thyroxinase is malfunctions or can't functions, you can see that DOPA concentration is high, but dopaquinones decrease and also melanin decrease. So therefore, again, it will result in the low skin pigmentation. But in this case, the albinism won't cause the mental or intellectual disability because this pH still function. It's quite unusual, both of them not functions. Okay, so you can see that thyroxine concentrations will be still high. Okay, thyroxine is high. Okay, DOPA is still high. Okay, can I see that? Okay, but the thyroxinase can't functions, therefore dopaquinones decrease. So oculocutaneous, oculo means eye. Okay, the eyes, cutaneous means the skin. So if you look at the person with oculocutaneous albinism, you can see that the eye pigmentation is low, and then the skin pigmentations can see that it's very low. So they have very, very fair skin, and also they will have the vision problem, reduced in sharpness, because our eyes, black color of the eyes because they don't absorb the light intensity. But now with lack of this, then it can create a multiple image blurriness in this case. Okay, so reduce the sharpness of the image. So ocular albinism can result in the mutations, okay, in several genes, but our syllabus here focus at thyroxinase only, a TYR gene, the thyroxinase. So the mutation in the gene for the enzyme thyroxinase, it may result in either absence of the thyroxinase or presence of the inactive thyroxinase. So therefore, the melanin productions will become low. Okay, so most common time mutation is because of single base substitutions, but we do have a frame shift as well. Okay, so albinism provides an example of study of relationship between the allele, enzyme, and also the human phenotypes. So what does this mean? Now look at this. If you look at the genotypes, big A, so they have the thyroxinase. Because they have the thyroxinase, then therefore, in terms of phenotypes, you will be able to see that, okay, this allele, this enzyme. So what is the phenotype? So darker, because of the melanin production. Same goes to the big A, small a, heterozygous. So heterozygous big A allele, both of them can code for thyroxinase, but with one copy, we still have sufficient thyroxinase. So therefore, you can see that darker skin. But an individual with small a allele, small a, so they won't have the thyroxinase. You don't have. So therefore, in terms of skin pigmentation, we'll have a low in the melanin production. So it gives us a different phenotypes, in this case, lighter skin. Can I see that? So this is how the allele, how we relate the allele with the enzymes and also the human phenotypes based on the skin pigmentation. Okay, based on the skin pigmentations. Okay. So the next disease here is the hemophilia, blood clotting factor. So hemophilia is a genetic disorder that impairs the body ability to make blood to clot. Okay, blood cannot clot properly. So the general one in our syllabus, we're going to follow with focus in this disease called hemophilia A, which have insufficient clotting factor 8. Okay, so these two I forgot. I guess it's just for information only. You can read. If you're interested to know the blood clotting mechanism, but generally, you follow these diagrams, you will realize that factor 8 actually involved here. So it's a cascade. It's a cascade activations one by one. So you can see so many factors. Okay, factor 8 involved in the activations of the factor 10 to become activated. When a factor 10 become activated, prothrombins will be activated into the thrombin. Thrombin is the enzyme that convert the fibrinogen, which is soluble, into the insoluble fibrin. Okay, so what will happen if, let's say, we don't have the factor 8? When we don't have factor 8, means factor 10 cannot be activated. So thrombins we won't have thrombin. Therefore, the blood clots cannot take place. The fibrinogens cannot be converted to fibrin. Without the fibrins, then you don't have the clots, no clots. Hence that. So it's a step-by-step process. Okay. Now, why we study with this disease? Because the factor 8 actually located on the X chromosome. It's X-linked. In this case, we call it as X-linked or sex-linked disease. Okay, hemophilia. So basically, if you look at for the male, one copy of the recessive allele can cause the disease already, because we don't have the dominant allele to mask the recessive allele. But for female who get the disease of hemophilia, then they must have 2 copies. But when a female, if a female is a hemophilic, they won't be able to survive long. The reason is because female, when it reach the puberty, you will have the menstruation. Can I see that? So means that a female with a hemophilia, they have to stop or to block the menstrual cycle. So means that the female cannot go through the periods of this menstrual cycle, because the bleeding will kill, excessive bleeding will kill the female in this case. Okay, so but for male, okay, you still can survive. Okay, because we don't bleed, and I have to make sure that we don't bleed. Okay, so as the hemophilia, the X-linked recessive disorder, female are very rare. Okay, female very rare. Okay, but for the male, then 1 copy. If the gene responsible for production of factor 8 or factor 10 present on the male X chromosome are recessive allele, then we don't have the equivalence on the Y chromosome to cancel it out. Okay. So how about the mutations? In this case, we have inversions, or we have the large base deletions. Those are the possible mutation. The inversion means upside down. Are you clear? Inversion basically means that, let's say ATCGCCAT, for example, we by right should read this way, but now they read it in the opposite way. That's called inversions of the gene, which is not in our syllabus. Okay, we only study 3: base insertion, base deletions, as well as the base substitutions. Okay, so I repeat again. So this disease, what we need to know here, how the allele actually functions. So the allele will cause the clotting factor. Okay, it causes the problem in the clotting factor 8. So without the clotting factor, then fibrins cannot be formed. So without fibrins, so blood clotting take place. And how in terms of mutation? So the mutation take place here, it may be due to inversions or large base deletions. It's not 1 or 2 base, but many base or nucleotides have been deleted. And then in this case, you can see that for male, 1 copy sufficient to get the disease, but female, they must have 2 copies to get the disease. So that's why it's an X-linked recessive disorder. Female are rarely have severe affected in this case. Okay. So we come to the last disease, a very famous disease, Huntington disease, because of quite a number of questions have been coming out for the Huntington, asking about Huntington. So Huntington disease is an inherited disorder, result in the death of the brain cell. So most of the time you can see that your mental ability decline into dementia. Basically, you forget, you tend to forget because your brain cell die progressively. Okay, so the disease is known by the autosomal highlights dominance mutations, where autosomal dominance means that you only need, a person only need to have 1 copy of the mutated allele to get the disease. Okay, so the gene is called as a Huntington gene, Huntington genes. So far, we do not know, okay, quite detail what is the function of Huntington gene. So some actually hypothesize that they are involved in the transcription, transportation, cell signaling. Okay, also involved in the microtubules. So this one all for your informations only. I won't go into the detail. If you're interested to know more, you can actually go and have a look. Okay, but basically what will happen here is the mutation involve a DNA segment known as the CAG trinucleotide repeat. So it means that it's addition. But it's not add 1. It continues to add CAG, 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 CAG. Okay, can you see that in this diagram? CAG, 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 CAG continuously. Okay, so because of this CAG, so normally CAG segment is repeated 10 to 35 times within the gene. You can see that this Huntington gene, we have about 10 to 35 times. Okay, so the person with Huntington disease normally will have more than 36 times, 36 to more than 120 times of the repeats. So people with 36 to 39 CAG repeats may or may not develop a sign symptom. So it means that it's not severe. Okay, 36 to 39, they have the Huntington disease, but they don't show a severe sign symptom. They may still survive well. But people with 40 or more repeat always will develop the disorder. So what is the problem here? What is the problem? Okay, now because of this CAG, 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 CAG code for glutamine, CAG code for glutamine. So when it code for the glutamine, what will happen here? When it code for the glutamine, so means that you will have this polyglutamine chain. Can you see that? And this protein lost the function. This protein cannot function now. So when it cannot function, what will happen here is our body do have the enzyme to cleave this protein, the defective protein, okay, behind this become a short fragments. Are you clear? So it means that you can see that we have the enzyme cut it into a very, very short fragments. Are you clear? But the problem is the glutamines, they are polar. So they are polar, they're going to attract the other protein and form what we call the protein aggregates. Can you see that? Protein aggregates. So these protein aggregates will interfere with the neuron function, okay, because this fragment can be misfold and form the inclusions body within the cells. So it means that you try to imagine this is a nucleus. We want to have normal functions, but a lot of this rubbish there block the function. Okay, we have this rubbish there block the function. So therefore, you can see that the cells actually lost the function, particularly the brain cells cannot functions already. Okay. But the problem here is, the main problem, you won't see, this is a progressive because accumulation, more and more accumulate. So it's a progressive disorder. What is progressive disorder? So it means that at a young age, a person with a Huntington disease may not know he or she has Huntington. Basically means that unless it's a family history, if no, they won't know. Okay, when they don't know, what happened here is they grow up and then they get married and then pass on this allele to the descendants. So when they pass on this allele to the descendant, again, as I say, that one copy of the allele can cause the disease already. So therefore, you can see that there is a scary part of the Huntington disease because it's the autosomal dominant disorder. Okay, it's an autosomal dominant disorder. Okay, so they only manifest or show the symptom when the person already reached the age of 30 to 40. Okay, so in this case, they already reproduce and pass on the allele to the next generation. Okay, so it is Huntington, and at this moment, no cure. Are you clear? Huntington disease, no cure. So we're going to look at this Huntington disease again in the next two topics when we look at how the genetics screenings take place to check whether a person have the Huntington disease or not. Okay, so with this, we have done. Okay, we have done the the topics for this